All right. Hi, everyone. Welcome to our last quantitative seminar this quarter. I am Zoe. I'm organizing the Quant Sem this year. So I'm going to start us off with a land acknowledgement. The University of Washington acknowledges the Coast Salish peoples of this land, the land which touches the shared waters of all bands and tribes in the Suquamish, Tulalip, and Muckleshoot nations. Our lab also acknowledges that we live and work on the ancestral lands of the Duwamish people, the first people of Seattle. I'm really excited to introduce our speaker today, although he probably won't be a new face to many of you. Trevor is a professor at the School of Aquatic and Fishery Sciences here at UW. He obtained his bachelor's in computer science and zoology and his master's in conservation biology at the University of Cape Town. And he did his PhD here at SAFS, working on the effects of catch-share fisheries on groundfish fisheries in 2004. He focuses on solving biological problems through data synthesis and mathematical models and works on a wide variety of research projects. His blue whale research spans everything from subspecies separation to abundance estimation and population modeling, including past assessments of the status of Antarctic blue whales and those off the west coast of North America. Trevor's happy to take questions as he goes, so people in person, feel free to ask your questions. Online folks, submit them in the Q&A and I will read them out loud to Trevor. Thanks everyone for coming, and thanks so much, Trevor.
Thank you, Zoe. And as she said, please feel free to interrupt. I'm from the old era when everybody used to just interrupt all the time and fight over the presentation, as opposed to the new era when everyone's super polite and lets the speaker finish and actually speak and things like that. So, and this, uh-oh, I lost it already. That's a bad sign. I'd ready to lose it. Oh, no. Come back. Thank you. This piece of work is very much an ongoing work in progress, even though I have already sent in 2 papers to the IWC and a third one is due on the 10th of April. So I really need to get this analysis done. And I'm hoping that when I get to the part where I'm explaining what I did wrong, that you'll tell me what I should do instead to do it right. So anyway, I really like this video because it shows a really cool behavior of blue whales for no other reason other than you can just watch it. There's a swarm of krill over here. Oh, okay.
Your computer may just fall asleep.
After 1 second. There's a swarm of krill approaching on the right. And what's fascinating is the blue whale appears to be able to track the distribution of that krill swarm, even though, like, how do you know where the krill are when you're underwater and you're like moving towards like a bunch of krill in the water? How do you know that the rest of the swarm is off to the right and around the corner? So if anybody can solve that, that's kind of a cool research project. But anyway, so what am I doing? Why am I doing it? And all that kind of stuff. So I want to know the status of each blue whale population. To do that, I need catch time series, I need abundance estimates, and I need a population model. And the problem is that for many of these blue whale populations, it's really hard to figure out how many were caught. Okay, we know the total catches, we just don't know which ones are caught out of each population. And the Indian Ocean is particularly problematic because there's so many different populations that live there, and none of them have their catches separated as they were recorded. So the plan is, I'm going to gather data from songs that blue whales make, fit a spatial smoother to that, and use that to infer where each population lives. And from that, I can use it to separate out the catches. So that's the plan. It's kind of weird. My screen here is not updating, but okay, I get the preview. So this gives you a big overview of blue whale populations around the world, or it would, if it lets me see it.
You can always project my screen.
But I just well, not showing again, but what if I just start this up? screen. Let's try again. So blue whales comprise of a bunch of subspecies. So northern blue whales are considered separate from Antarctic blue whales in blue, probably Chilean blue whales in green. And for those that are colorblind, I apologize, it's everything within this region here are pygmy blue whales. Maybe we should just project from your one.
Yeah.
Yeah, because this is not going to be great. It just keeps on turning off on me. Sorry, everybody online. We are going to project from the other screen.
Do you want us to just screen share or you want me to screen share?
Well, I don't know if it's, why don't you screen share as well? If you just click on the project, yeah, that one done.
That work?
Yeah. Check on that thing. Okay. Yeah, go one up.
There we go.
So today what we're going to look at is this set over here of Antarctic blue whales versus pygmy blue whales, and how to separate out these catches here. These ones here are a bit easier because they're bigger animals, you can detect them differently. And the key is the way we know they're all different populations, and all of these letters here, the CN, CWNPO, SWPO, SEIO, SWIO, all those combination letters are what we think of as a separate population of blue whales. How do we know that when we can't tell them apart at sea? Is because they make these unique song types. And I'm trying to advance my slide on this one now. So if I played these songs to you now, you wouldn't hear anything. They're not quite inaudible that they're so deep, but you'd need a really big subwoofer, not like something like a laptop speaker. And then the room would kind of vibrate a bit, and you'd have to listen and be like, oh, okay, was that something? So typically when you want to listen to them, you speed them up 10 times, and then you can hear what they sound like. But as you can see from these 6 different song types, they're quite different. So this is over time, you can see that this is like 6 or 7 minutes. So repeated song is like 1, 2, 3 units for this population, 2 units for this population. Antarctic might be 7 or 8 units in 7 minutes, but they're super long, super low, super deep. And one of the cool things about these blue whale songs is that they are getting just slightly deeper over time every year. Every population in the world, no matter what their population status, no matter how much boat noise there is, no matter what the temperature trends are, where they are, they're just all getting a little deeper every year by a little bit of a different amount in each population. And no one really knows why. So if you have a good explanation for that, that's another research project. But what we'll use them for here is this diagnostic of which population is which. So if you can go and you can put a hydrophone in the ocean and listen for whale song, you could figure out which population those whales belong to. We think that's the working hypothesis. Okay, but let's take the first step. How do we distinguish between the big blue whales and the little blue whales, the Antarctic blue whales and the pygmy blue whales? And the answer is they have different lengths, as you might expect from the names. And the key piece of information here is that pygmy blue whales max out at about 80 feet, 24 meters. So Antarctic blue whales only become sexually mature at just under 80 feet, and they can max out pretty at about 100 feet. So something close to 30 plus meters. And what you can see here in the data is each of these little histograms is the length distribution of catches taken at a particular latitudinal and longitudinal bin. So this is 150 to 180 east and 70 to 76 south. And you'll see that some of these, almost all of the catches were over 80 feet. So the red bars are over 80 feet and the gray ones are under 80 feet. So visually you can see that, hey, of all of these sexually mature female blue whales, pretty much I can draw a line that says these ones were Antarctic blue whales and those ones were pygmy blue whales. So that step is pretty easy. I can do that by just looking at lengths and saying, well, here's the boundary. These are Antarctic, these are pygmy. Let's move on. Step one, easy part. Okay. And the same is true if you zoom in even more. So here is 1 degree latitude by 10 degree longitude bins. And you can see there really is like a very distinct separation in terms of space. I don't know why there's such a separation in terms of space. They're both equal. They just, for whatever reason, prefer to be in different places during summer when most of the catches were taken.
They're seeing the presenter view, not the full screen slides.
Okay.
So let's
Just stop share and share again. Maybe you say share the screen. Yeah, share desktop too.
Okay.
Maybe that'll work.
All right.
Hopefully that. Can they hear fine?
Yeah.
Okay.
Oh, here, go back there.
Okay, so let's zoom in now on just these pygmy blue whale regions. So all of these red dots are pygmy blue whale catches. And if you look really closely, you'll see that there's not a perfect separation. So every now and again, there were catches made that were identified by the whalers as being pygmy blue whales. So there are a few red dots on this side of the line. And there are a few blue dots on this side of the line if they were bigger than 80 feet. So using that length discrimination. But we can now focus in on this area here and ask, how do we separate out these different populations? What I can say is the New Zealand population, the Australia/Indonesia population, the Madagascar population, the Central Indian Ocean population or Sri Lanka population, and the Oman population or the Northwest Indian Ocean population. So how do we do this, and how are we going to figure this out? And the one point we don't know much about is Durban. So there were thousands of blue whales caught in Durban. We don't know if those were Antarctic or pygmy blue whales because the whalers didn't bother to measure how big they were. There was just too much effort. And I should say that whalers in general were pretty lazy about measuring the length of blue whales. So you would think that it wouldn't be too hard to measure them somewhat accurately, at least say to the nearest foot. That wouldn't be too hard, but it turns out that a lot of the times they just couldn't be bothered and they were just estimated to the nearest 5 feet or 10 feet. Looks like a big one, let's write down 85. So there are definitely issues with the data. For Durban in particular, we have a hydrophone that one of my collaborators, Fanny Shabangu, has deployed off Durban to listen for songs there, and we hope we'll get data pretty soon, like within a month or two, that'll tell us if that's mostly Antarctic song or mostly probably the Southwest Indian Ocean song. So that's our hope. We'll at least clear up those set of catches there. But for the moment, we're going to set that aside. Yeah.
Is the district, well, I guess this plot works. Are the dots roughly the distribution of the fish, or is that really just indicative of where the fishery is operating?
For Antarctic blue whales, the whaling industry was very much whaling everywhere. And so it's very indicative of where the whales were. For the red dots, most of the whaling happened in the Antarctic where there are lots of whales. And then only as those became depleted did they start looking elsewhere and discover, like, oh wait, there's another whole population of blue whales, another subspecies that's smaller. And so that's many fewer expeditions. And so I can tell you exactly where they're from because the Soviet Union basically came through the Suez Canal up there, headed out, whaled wherever they found blue whales, whaled down, got to Antarctic, did everything else. You know, then some years they went up to New Zealand after they finished whaling, and then they went back past Australia and up. Some years they came out this way, whaled down as they came down this way, and then across and down. The Japanese whalers originally discovered them pretty much whaled exclusively down here. So this pretty conflated with how much effort there was. Yeah.
What years are these data?
These are all 20th century whaling records. The Antarctic blue whales started in 1904.
Okay.
The pygmy blue whales largely started in 1958 when they discovered them. So there's a temporal time. Yeah. And I should say that if you're interested in plotting or examining the data in greater detail, we're working in a collaboration with a couple of computer scientists at the Paul Allen School of Computer Science and Engineering. Maya Patel is producing a really, really nice visualization of all these catch data, where you can drill down into the data and then ask to extract data, look at it in more detail, look at it by species, by year, by sex, by pelagic versus land stations, all kinds of things like that. And the ultimate hope that I have for that is we'd be able to link together all the whaling voyages to figure out where there was effort and where there wasn't effort, and use that to correct the catches for some kind of catch per unit effort, like where the whales actually might be. So that's ongoing. It's really cool. They have skills I don't have, which is true of almost everybody. So the wrinkle in all these catch data is this. In the 1950s, at the point where the whaling industry was starting to see the end, because blue whales were down at low levels, fin whales were at low levels, humpbacks were banned, right whales were banned because of low levels, the Soviet Union started building these massive factory fleets. So each of these boats here is what's called the factory vessel, which processes whales. Each of these would have somewhere around 20 catcher vessels that would go out looking for whales. When they found any, they would all converge and catch them, bring them back for processing on the back of one of these vessels. And these were the biggest factory boats that have been built, biggest fleets that have been built. They were built starting in the late 1950s. And the rest of the countries who were whaling were thinking, how are they catching enough whales to make this economical? And to some extent, that was because the Soviet Union didn't care that much about making a profit. They wanted to produce raw materials and so on, but also because they were doing some minor things like ignoring the regulations on species. So they were illegally catching blue whales, right whales, humpback whales. They were illegally ignoring the regulations on latitude. You were not allowed to do pelagic whaling north of 40 south, this red line. And you'll notice that there were some whales caught north of 40 south. They were illegally catching whales out of season. So blue whales could only be caught basically in January and February and at the start of March. They were catching them starting in October, November, December, all the way through to May. And they were ignoring all other regulations, like you weren't allowed to catch whales that were lactating, you weren't allowed to catch whales below a certain size. They just ignored everything and caught whatever they found. Okay. So part of the problem with this is they were not reporting all these catches at the time. So there's this, for many decades, there was this big gap in the data. And only with the overthrow of the Soviet Union, perestroika, glasnost, all those things, did those records come out. But not all of them have been recovered. So it just depended on whether the whaling biologists happened to keep the truth, like in the backyard of their shed, waiting for the day they could release them, or whether the data were lost altogether. So we have some data that are not perfectly known. So all that. So those pygmy blue whales were discovered late, mostly caught by Japan and the Soviet Union. There may have been some from land station bases, but most were all caught by these open ocean big expeditions with all the whaling vessels going out and so on. Issues that some are missing location or month. We think we have the right number that were caught every year. We may not know exactly where each one of those whales was caught, but it's like at the level of 90 to 95%. So we have individual locations for each whale for the ones I'm going to talk about now. And then this final thing that if you're really interested in length data, the problem was that the legal minimum length was 70 feet. And most pygmy blue whales are not 70 feet because they're small. And so what happened for some countries is if they caught a 65-foot whale, the biologists would be like, Looks like 70 to me. Then right down 70. So if you look at the length distribution, it goes nothing, 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 nothing. Massive peak at 70 feet. You know, goes down again, nice normal distribution, the end tail of a normal distribution. So, you know, you have to take the length data with a grain of salt for some countries. Okay. So what do these catch data look like? What are we trying to separate out? So these data, I'm going to show you October, November, December, January, and another one with the other months. The red dots here are Soviet Union catches. There are no, well, there are a couple of purple dots over here for Japan. Remember, the whaling season was January onwards. So October, November, December, most of January is all illegal catches. And the green dots are scattered catches that other expeditions made over the course of many decades that happen to be in the right area. As we get to February, March, April, you can see the Japanese catches, the purple dots coming online. And you can also see how the Soviet whalers would have gone down to the Antarctic and then come circled back past Australia and New Zealand, catching whales off the coast of Australia and New Zealand before they headed back up to their bases. So that's a sort of spatial and temporal effort. But it does mean that if the whales are moving from month to month, we have to take into account where they are in particular months, because the catches were in different places in different months. Okay, so we have these songs. The next question is, can we figure out where each of these populations is producing the songs? And the answer is, we can if you go and beg, borrow, and steal, and ask a whole massive list of people if you can have all the data that they spent years collecting. Will you just give it to me? And the answer is that in this community, everybody's very, very willing to share because they can see the value of a bigger goal. So each of these lines represents someone's research project that they spent likely years collecting the data and also years analyzing the data to get information on, by month, how many hours were analyzed and how many hours of each of the populations were seen. And also, in some cases, they just looked at days. So how many days of data were collected and how many had observations. The question?
Yeah. Someone's asking in terms of whale stretching, what were the countries who weren't whale stretching doing? Were they listing the accurate length but harvesting the whale anyway, or listing the accurate length and discarding the whale?
In the context of this project, the Soviet Union were actually recording the correct lengths, but not sending those data to the IWC. So when the correct data was reassembled, those were the actual correct lengths. So you can look, and there are lots of whales caught under 70. For Japan, they were much more inclined to say they're all 70 and submit 70 feet as the length and not have what the actual lengths were written down anywhere. So those can't be retrieved. None of them were, when the Soviet Union was misreporting, they were doing things like saying, we caught 2 fin whales of such and such a length, instead of saying we caught 1 blue whale, because a blue whale is about the same amount of oil you get from 2 fin whales. So the misreporting was, in some cases, they were recording more whales caught of some species than they actually caught, in other cases less or none. So there's a variety of ways you can try and cover up, but not very well. Okay, so one of the problems with the data, and here I'm going to go on a great digression for those who are quantitative. So I apologize to all the whale people, but this is going to be a digression for the quantitative people. So one of the problems is we have some data where they looked at a certain number of days and said, we looked at 100 days, and of those days, 30 days included calls from this particular population. In other data sets, we have records in hours. So we went and looked at 2,000 hours, and of those, 300 hours were recorded. And of course, those aren't equivalent, because if you just heard 1 call in a day, there would be 1 out of 1, but for hours, there would be maybe 1 out of 24. So the 2 data sets are not quite equivalent. And so to merge these together to do the analysis, I had to figure out how you convert them. And luckily, some of my collaborators, like Gavrilov and Macaulay, have this huge data set where everything is recorded by hours in a consistent way. And so you can subset the data either to talk about it being by hour or by day. So you can do both ways. And then each of these dots is 1 month of data. So 28, 30, 31 days of data on the x-axis, proportion of days with records. And the y-axis would be proportion of hours within that same month. So each dot is 1 month of data. As you can see, there's a ton of locations, a ton of hydrophones over a ton of years and a ton of months that these data are based from. And the question is, what do you fit to this? And how do you get a good fit for a model to these kind of data? It's bounded, the y-axis is bounded between 0 and 1, the x-axis is bounded between 0 and 1. And I tried all kinds of things. I was like, well, what about an exponential? But that can go greater than 1. You know, what about, tried all kinds of things. And eventually I gave up and I posted on Twitter and got the hive mind to solve it for me. And Gavin Fay, who was a PhD student here, Andre Pants, said, why don't you try the cumulative distribution of a beta function? Okay. And use that to predict the mean fit. So the cumulative distribution, remember, cumulative distribution goes from 0 to 1. Okay, a beta function is bounded between 0 and 1. So the x-axis is bounded between 0 and 1. The y-axis is the cumulative distribution. So that's going to be between 0 and 1. And it's going to be increasing because it's the cumulative distribution function for those that have done these stats. And it has all these great shapes. So as you change the two parameters of a beta function, the A and B parameter, alpha and beta, you get all these great shapes. So you can just estimate what is alpha, what is beta, that gives you the best model fit to your data. Kind of cool. I thought that was kind of cool. Okay, and your predictions, it's just literally this function in R is the prediction is P beta, which is the cumulative distribution function for beta function. With alpha and beta, search over alpha and beta, find the best fit, and you're done. Except that what likelihood do you use around each prediction? Okay, you predicted the mean value, but there's uncertainty around that mean that very clearly ranges in kind of a weird way. So how do you go from one to the other? And your prediction is, it's in binomial format. So each of these data points is something like, say, 720 hours, of which it was 20% of that would be 140 or something like that, hours with detections. So it's a binomial data point. But with a sample size of 700, the binomial distribution is super, super narrow. Okay.
Trevor,
Yes.
I guess I don't buy this fit because,
Oh.
so like when x-axis 1,
Yeah.
it's by definition your model, y-axis is 1 as well. But like there's like a ton of variation. So like what's the mean of the data when the x-axis is 1?
Yeah, so we actually, what I've actually done is I've carved off that as a special case. Because even though 100% of days have detections when it's 1 on the x-axis, it doesn't mean that 100% of hours have detections on the y-axis. And so I've actually shifted everything over just a little bit and only made predictions up to 0.99. It's kind of a...
It seems like you could just add a third parameter of scalar plus 1 and just multiply the CDF by it.
Yeah, so there certainly are other ways of doing this. I mean, or another way of doing it is just to say, let's take the ones where there are 100% of days and just treat them separately and just use the average of those ones as your prediction or something like that. Yeah, I'm not saying it's perfect, but it's done. So if we assume that our predictions are binomial and your sample size is 720 hours, this is what you'd expect the likelihood to look like. You'd expect 95% of data points to fit within these confidence intervals. That's just a property of the binomial distribution. So if your prediction is 10% and n is 720, then almost everything's going to be between like 9 and 11%, something like that. our data doesn't look like that at all. And so the next part of this puzzle is, so what likelihood does work? You're not allowed to tell me because you told me already. Anybody know what likelihood you might use for binomial type data that are over dispersed?
I know.
I might have told you already. I didn't know either. You can use a beta binomial. So this is a more general concept that when we look at ecological data, very often the tails are fatter than you'd expect from a standard statistical distribution. So we're used to thinking about everything is normal, we can use a normal distribution for everything, and then you go and look at your ecological data, and it turns out that you find more things out in the tails than you'd expect. And so we have terms for that. We call them outliers, or black swan events, or unexpected things, or surprises, or whatever, because they happen so often that we, in real life we have terms for those. And so if you want a distribution that sort of looks like a normal but has fatter tails, you can use a t-distribution. If you have a distribution that should be Poisson, like whole numbers generated, so on and so forth, and negative binomial has fatter tails. If you have a multinomial that doesn't fit the assumptions of every data point being random and independent, something like a Dirichlet multinomial is a way of getting around that. So we use those for fisheries catch at age data. And for a binomial, you can use a beta binomial. And all these things, the left-hand side is nice looking distributions and the right-hand side is nasty looking distributions. That's a good general trend. But also, my thesis is that almost everything we deal with in fisheries and ecology should be modeled by one of these things on the right. It's very, very seldom that it really does meet the assumptions of the things on the left. Unless we're dealing with means, and then we can rely on the central limit theorem. Okay, so that's the binomial. Nice looking, and for those who don't think that's nice looking, I apologize. Beta binomial is nasty looking, okay? And there are different ways you can parameterize this. That's not the only way you can parameterize it. That's just the nicest looking way, okay? And not only that, that gamma thing there is itself a nasty looking function, okay? That in itself, all these gamma things here is another nasty looking function that you can think of as a generalized factorial function. So it's very closely related to factorial, except it works for non-integer numbers. So factorial has to be whole numbers. The gamma function can work with non-whole numbers, and there's a +1 or -1, like a gamma of n is the same as a factorial of n-1, something like that. Okay, but luckily for us, we can just call dB binom the density of the beta binomial in R and get the density of a beta binomial and not have to worry about actually coding that up, which could be annoying. But if you did have to code up, there is a gamma log function in most programming languages that lets you do that. How are we doing on time?
106.
So if you're doing binomial, as your sample size goes up, the distribution gets narrower and narrower and narrower. Right? So if we were doing on the x-axis here, n is 30, so this would be 30 over here, and this plot n is 100, so 100 would be over here. If n was 720, like our data, we would be able to get and have this very narrow distribution. But if we go and look at a beta binomial, you have one extra parameter can be parameterized with one extra parameter, an over dispersion parameter. Here I've called it m, or it could be sigma. And as m gets either bigger or smaller, depending on how you parameterize it, the shape of the likelihood gets more spread out, which is what we want. Okay. So this would be if m was, I've done this with m being big, being like a binomial. As n gets smaller, it spreads out. Could be the other way around if you just parameterize it as 1 over m. Then you can think of it as like a sigma, like a variance parameter. And that means that we can fit our model and have all the uncertainty done. Okay. The only reason I talk about this now is because I'm going to use the beta binomial later as well. And now at least you know what all this does. And now you have a new distribution that you're going to think of every time you have binomial data. You're going to think, but wait, maybe this is beta binomial. And then you can go down the same rabbit hole I went down. So you're welcome. So having done all this, we can now convert all our data into hours. And if we wanted to include the uncertainty and all that, we can use the uncertainty around this prediction in our data and make our data uncertain as well. It turns out it makes almost no difference, including all the uncertainty or not, that the mean gives you the same answers. But anyway, it was fun doing it. Okay, so now we have our data. What do the data look like? And is this amenable to answering the question, which is where do each of these populations live? So here are all the data for the Northwest Indian Ocean, the Oman call type. They're not detected in very many places. So each of these gray circles is a place where a hydrophone is put, and they didn't detect them. Any circle with a black outline, they detected that song type. And where the circle itself is filled with dark shading, there were lots of detections at that place. So any circle that just has a circle with no shading in the middle was pretty just a couple of detections in a few hours, and not detected anywhere else. So this is going to be a little tricky to fit to because some of these months have very few data points, very few actual detections. The Central Indian Ocean, so you can flip backwards and forwards. You can see there's the Northwest. Here's where you hear the Central Indian Ocean, the Sri Lanka blue whales. So you can see there are quite a few places where they do hear them a lot. So almost every hour, the calls in this month of them. You can see a little bit of movement up and down. Here are the Southwest Indian Ocean, lots of places where you hear them. So you can sort of see the different places where each population occurs. And the Southeast Indian Ocean, these are the Australia and Indonesian population. And finally, the ones off New Zealand are very constricted to just a few places. Mostly they seem to be in New Zealand year round and found very few other places. And just for comparison, here's where you hear Antarctic blue whales. And one of the deep mysteries of blue whale research is, where do Antarctic blue whales go in winter? So the winter months here are June, July, August. And the answer is wherever they want, right? Pretty much everywhere. But even more confusing, we always have this conception that Antarctic blue whales are all feeding down in the Antarctic in summer. So that's January, February, March, where they were all caught. But if you look in January, February, March, you can hear their songs across a pretty broad range of the ocean. And I'm not even showing the full data set all the way with the Pacific and South American song, but you can hear Antarctic blue whales basically anywhere in the Southern Hemisphere in any month of the year, but more so in the winter months. And remember, there are only a few thousand of them, we think. So it's kind of confusing to me how spread out they must be for us to hear them almost everywhere in almost every month. They are very loud compared to the other ones. Okay, so what's the method we're going to use? And I'm going to show this figure from Cole's paper because he did such a good job of this that I thought it would be easy to just transfer it over to the Indian Ocean, not realizing just how much work he did and how much work I would try and do to take over when he started working on this project and said, no, you can do the rest of it. It's easy now. and maybe for him, not for me. But his idea, he did the same kind of thing for the North Pacific. So there are two song types, the Western and Central North Pacific song type in red, the Eastern North Pacific song type in blue, and one divided by the sum of the two gives you the probability of hearing one or the other in each location in the ocean. And you can use that to separate out catches, and here are the catches of the Western North Pacific, Eastern North Pacific population. So that's the general idea we're going to use here. And the method for doing this, and this is where I went horribly wrong, is using the GAM LSS package. So the generalized additive models for location, scale, and shape. And the nice thing about this package is it's super flexible. You can do GLMs, you can do GAMs, you can fit models with binomial data, with Poisson data, with normal data, with beta binomial data, if you want. And the reason to think about using this package is that not a lot of packages allow for the beta binomial to be used as the likelihood function. Our data are binomial format, so at each location we have number of hours of wells were detected versus total number of hours. So that's like flipping a coin, how many times is it heads, how many times tails. But because each hour is not independent, if you hear a whale now, are you going to hear a whale 1 hour later, 2 hours later, 3 hours later? Probably. So that you're going to have more high numbers and more low numbers than you expect. And also because the hydrophones themselves, the data collecting devices differ. So some of them are what's called in the sound channel, where you can hear whales from far away. Some of them are in shallow waters, you can only hear whales that are really close. And so the detection range of the hydrophones is different, which means that the data themselves are not independent and random collections of information.
Someone's asking if you know what the average detection range of the calls is.
There is debate. If you ask some people, they will say you can hear individual blue whales for many hundreds, perhaps thousands of miles. If you ask other people, they say you can only hear individual calls from maybe about 100 kilometers away. So there's a pretty wide range. It does depend on bathymetry. It depends on where the hydrophone is placed, depends on how loud the calling animal is. There is a band of like the Antarctic blue whale chorus. If you put a hydrophone anywhere and you listen, you can't hear the individual Antarctic blue whale calls, but there's like a band of noise at that frequency that is like Antarctic blue whales that you can hear throughout the entire southern hemisphere. These calls we're using are ones where you can hear the individual calls, not that chorus. Okay.
Cool. Yeah.
So the general idea here is we're going to fit something that predicts the mean probability of detecting a blue whale based on longitude, latitude, and month. And that CS stands for a cubic spline. So that's a GAM way of doing things where you can have a more flexible, smoother fit to the data. P is the probability of detecting a song in an hour. And sigma is that over dispersion term in the beta binomial. And probably I shouldn't be trying something as complicated as that. I should be doing something simpler, and that's part of where I went wrong. We can also test very easily different types of models, a binomial versus beta binomial, GAM versus a GLM, the linear model versus the cubic spline fit. In Cole's paper in 2014, the beta binomial was at least 12,000 AIC units better than the binomial. And if anyone knows how AIC works, if you're more than about 10 units better, you're definitely better, and you can discard the other model. So my presumption was that the beta binomial would be what I would use for all these model fits. And to implement it, it's literally like this. There's a bunch of complicated control files and stuff, but then you give it a data frame that contains number of hours with detections, number of hours of not detections, so the two sum up to the sample size. You tell it what you want your fit to be, so cubic spline, longitude, latitude, and month. What your formula is going to be for that over dispersion parameter, the sigma formula, which could be simple. What your likelihood is going to be, the beta binomial, and what your data is, and then you give it those control parameters that help it find what a model fit. And you might have to do this a few times to make sure it converges. And your data just look like this: latitude, longitude, month, number of detections, number of hours of no detections, and which population you're looking at. The nice thing about the GAM LSS model is you can very quickly and easily do things like change that entire thing to a binomial by just changing the likelihood. So instead of beta binomial, change to binomial. Or if you want just to estimate one over dispersion parameter, instead of saying the over dispersion can vary by latitude and longitude and month, you can just set that to 1 and it'll just estimate that one parameter. Or if you want to change it to GLM, you can change this instead of saying cubic spline, you just say it's a function of longitude, latitude, and month. So it's a really nice package and allowing you to look at different types of models. Okay. So what do the fits look like?
10 minutes, by the way.
10 minutes to go. Okay. The problem I had, and I was basically fitting these things wrong, is that I couldn't get the beta monomial to fit for some of the populations. So this model is a binomial fit. And the thing with a binomial fit is it wants to say 100% of ours in the middle are detections, and everything just outside that is a 0. Right? So that one, I could only get a binomial to fit the way I was doing it. Whereas these other ones, you can see how much more spread out it is as soon as you set up a beta binomial, because it allows these fatter tails from further away. And it doesn't say there's 100% detection in the middle. So there's your top left, your Central Indian Ocean, your Madagascar population, your Southeast Indian Ocean population. And each of these things misses something. So for example, the Australian one, we know from satellite tag data that they migrate around the coast and go up to Indonesia, but none of these model fits can produce that kind of prediction. So there's a question about whether you should try and include other forms of data in the model fits as well.
What is the
Oh, so shading. So dark, the shading is actually big circles are high probability of hearing the whales, and small circles are low probability. And no circle is very low probability. So we know from satellite tag data they go around there. Yeah.
I'm wondering the fact that you have like big patches, so that very fine patterns.
Yeah.
That's what we're searching when you do migration. Not really key. Like the fact that you're working on splines, and spline make big patches. When if you were working on like spatial random fields, social random fields, you may like better evidence these fine scale patterns. That's suggestion.
Yeah, and that's certainly true. And it's kind of an open question given the data, the kind of data you have. So given the data you have, if you fitted with more complexity and allowed it to have finer nuance, what would it say about this area in the middle where there's no data? Would it then say there's no whales there? And would that be realistic? And in some sense, for the purpose of catch separation, I'd rather the model said that these ones occur here than say they occur here and here, but not in the middle.
Yeah.
In the middle, there could be some other population we haven't detected there either. It could be there, but that doesn't make as much sense to me.
But then by replacing splines by random fields, roughly you may see that.
Yeah, certainly. I mean, my lack of knowledge in this field is the major drawback, because I know there's so many ways of fitting spatial data, and I don't know what the right way is. I do know a lot of methods that are out there are not very good at handling beta binomial type data, so that's been a major constraint.
Perhaps you need, for instance.
Yeah.
You could like model these to random fields instead of splines.
Yeah. So that's certainly an approach that I would like to explore on that. Yeah. Cool.
Yeah, I mean, I agree. I would definitely try it. And my intuition actually is that you would not get fine scale patterns, because the effect of the over dispersion of the beta binomial is essentially downweight the data. So you don't have that much data to drive, I think, really large fine scale changes. If that makes sense. So I kind of think it's going to give a similar, more broad scale pattern, spatial, like the gas in the field would. But we need to try. We should try it.
Yeah, first try. You might do it.
That's where I put my money, though.
Yeah. If anybody wants to work on Inlet with us, happy to. Yeah.
And if I understood the way you set the model, month here is entering as a fixed event, right?
It should be. That's one of my errors. I haven't actually run the model with month as a fixed effect, but when I rerun it, yes. And that should solve some of the issues.
Yeah, that's where the mean is actually auto regressive, right?
Yeah. Yeah. So if I get time, I'll tell you all the things I did wrong. But here's the New Zealand population fits and Antarctic. But this is what it really boils down to. that you take each of these fitted surfaces, which gives you a probability of hearing the whales at each location. And then you can turn it around and say at each location, what probability is it you would hear each of the population songs? And use that to allocate the catches. So this is the result in some sense. And it has problems with it because of this mixture of some models that were binomial and some that were beta binomial. What you get is in the middle of the predicted binomial places, it says 100% totally Northwest Indian Ocean. But go just a little bit outside and you get that red square at the top left, where that's just a little outside where it says that should be, and a little outside of guess what is where there's like a very small probability it could be the central Indian Ocean population. And 0 versus a very small number says, oh, they should all be central Indian Ocean. So there's this problem that I have if I can't get all the beta binomial models to fit, that it is doing the what probability is wrong. And the same thing occurs down in a lot of other places. So like these are the Australian population. They're so spread out that there's some probability that even these ones in the central core out here, it's saying could be Australian whales, even though we don't hear them out there. But the model fit is spreading it out so much that it's saying some of these could be Australian whales when they really aren't. How much more I want to say. I'm just going to quickly go to this thing, which says basically that some of these populations at the end have very large catches taken out of them. So it's likely that 2 of these populations are quite depleted. Others of them have very small catches taken out of them. So the New Zealand one, we think there are at least 600 or 700 still living there now. So 400 are taken out, they're pretty fine. So you can see how having the total catch time series and a bit of abundance estimates, you could then figure out what the status of the population is. Big catches, small numbers, they're depleted. Small catches, big numbers now, they're probably not depleted. And that's the next step of the project. I'm going to stop there and give us like 5 minutes of general questions if anybody has questions or anybody online. Yeah.
Going way back, but I'm just curious if you know how much the songs, you said they were decreasing in frequency every year. Do you know by approximately how much that decrease?
Well, when I started doing this kind of stuff, they were saying that the Antarctic song was 28 hertz. It was very distinctive. It's 28 hertz. Now it's 26 hertz, maybe lower, maybe it's 25 hertz by now. So over the course of 2 decades, it's gone down by 2 hertz. And that's pretty typical. There is another call type that we're not sure what species makes it, but it has gone down even more rapidly, like 6 or 7 hertz in the space of 10 years and then jump back up again. We don't think those are blue whales. There might be right whales. They call them the spot call. So maybe blue whales will go down, 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 down until it's too deep for them to make the calls, and then maybe the whole population jumps back up again to some high number. I don't know. Yeah.
Does the detection probability for each individual species at a hydrophone depend on how many other calls, how many other species or not species, populations are detected in that area? So, because is it more difficult to parse out calls if there are more populations detected in one place?
It depends how many individuals are calling. At some point, it becomes hard to detect individual calls from even the same population because they're all sort of overlapping. But generally, it's easy to see if there are multiple calls of different populations, if they're like just 2 or 3 calling. Where it gets hard is if there were 50 calling, you wouldn't be able to detect individuals. But yeah, you can generally have multiple calls from multiple populations at the same place. Yeah.
I'm always wondering with the kind of data you're using, how much the sampling bias affects the results
A lot.
for showing, because the sampling is not like standardized, so for holistic sampling or technical one.
Yeah.
So this would strongly bias the results or at least modify the results.
Yeah, absolutely. Yeah.
That's-
Yeah.
That's a big challenge, like account for it in difference. So,
Yeah.
Have you some thoughts about it or?
Well, what Cole did when he did the North Pacific was trying to get an idea of the uncertainty in your predictions. So if you didn't have certain data sets, what would it look like? So the next step is to do bootstrapping. So if I can get all these models to run automatically and all converge, which I was having trouble with last time I looked at this, then you could randomly resample from the data sources. So some would get sampled twice, some would not get sampled at all. Refit the models, give you a prediction, resample randomly, refit the models, get a prediction, and that would give you some idea of the uncertainty in your predictions. It would give you at least the ranges of how the catch time series separation would shift. It doesn't, you can never really get around the problem of the data not being perfectly like randomly allocated. And each of these data sets is, some of them are quite big. So there's a French data set called Oasis Bio that's like most of the ones in the middle of the Indian Ocean. They have like, I don't know, like 20 or 30 hydrophones they put out there. They've been out there for years. Every year they go collect the data. When there's one around Australia where there's, I think 8 or 9 different locations around Australia. But a lot of them are just like a boat going along and like, hey, we want to see if there are any whales here, and throwing a hydrophone off the back, hydro buoy, that like the same things they use to find submarines. So there'll be like 1 an hour. They just throw them off and like listen until it gets too far away. That's pollution. Okay. But so they get these just a point for an hour at a time as they go along their track. Or they might go around all the way around the Antarctic, just doing that every day or two, dropping a hydro buoy down. So super different types of data. Yeah.
We should probably wrap up there.
Thank you, everybody.
But yeah, thanks everyone. Over.
Sorry, went over time.
You didn't. You were exactly on time.